Hi friends, welcome again. Part 4th from reproduction in lower and higher plants. So let us start pollen-pistil interaction. After pollination, the dusted pollen grains or received pollen grains on stigma will get germinated. That is, they will form pollen tube, and there are chances of deposition of wrong pollen grains. Means they may be from other plant, they may be from another different species belonging to same plant. So such pollen grains automatically get discarded from pistil, and these events, that is deposition of pollen grains on stigma to the entry of pollen tube into ovule. So these events are said to be pollen-pistil interaction. Okay, so here pistil recognizes the correct pollen grains and allow them to enter into style, and they will reach up to the ovule. Here in diagram you can see this is nothing but the pollen grain which has formed pollen tube, and this pollen tube is entered within the ovule. So compatibility and incompatibility of pollen pistil is determined by a special proteins. This process involves pollen recognition followed by promotion, means it can allow the further procedure, or inhibition of pollen grain. Compatible pollen absorbs water and nutrients from the stigma and germinate to form pollen tube. Already I have told you. Now this pollen tube will grow through the style and it will reach into ovule, and it will come in contact, or it will enter within embryo sac. So the tip of pollen tube enters in one of the synergids and then ruptures to release the content. In further diagram we will study that one. Due to pollen pistil interaction, intense competition develops even in the compatible pollen grains, means they will start competition with other pollen grains which are in race. So this pollen pistil interaction plays an important role in sexual reproduction and again the formation of seed. Pollen grain can also be induced to germinate in synthetic medium. They can be germinated artificially in synthetic medium. Sucrose is the medium where the pollen grains get germinated and they can be grown in vitro. Even this sucrose along with the boric acid accelerates the pollen germination procedure. So by artificial method also we can dust pollen grains on desired stigma, that is called as artificial hybridization. This is one of the major approach which is used in crop management or crop improvement. So here we can only use, or we can only take the desired pollen grains, and these desired pollen grains will be dusted or pollinated on desired stigma to carry the fertilization, and it is carried by two way, that is nothing but emasculation, which consists of the next procedure, that is said to be the bagging. Here in diagram you can see emasculation. The forceps is used to remove the unwanted material, that is if the flower is bisexual, then it can be converted into unisexual, or you can say female flower, by removing Here you can see the androecium, and now the flower will become only female flower, okay, with gynosium only. So why it is carried out? It is carried out only to carry the desired pollination. So it avoids the self-pollination. Emasculation is nothing but removal of anthers well before anthesis. Okay, and further, after dusting the desired pollen grains on this gynosium, it get bagged. Next is nothing but double fertilization. Double fertilization is discovered by Navskin in liliaceae plants like Lilium fritillaria. So this is a unique, you can say unique thing which is only found in angiospermic plants. So let us see the definition of double fertilization. Double fertilization is nothing but the fusion of first male gamete with egg and the fusion of second male gamete with secondary nucleus is called as double fertilization. Okay, so what happens here, when the pollen grains get germinated, it will form the pollen tube. Okay, from pollen tube or the pollen tube consists of two male gametes and they starts to travel through pollen tube, and the movement of such male gametes is said to be the siphonogamy. So here you can see easily they have reached here in diagram, they have reached up to the or into the ovule, and they are now going to enter in embryo sac. Okay, so what happens here, entry of pollen tube may be by three directions. If it is taking place through micropylar end, such entry is said to be the porogamy. Here you can find. If the entry is from chalazal end, then such entry is said to be chalazogamy. Sometimes they may enter by piercing the outermost covering of embryo sac. You are known about that. That is nothing but integuments. By piercing the integuments, such entry is said to be the mesogamy. So here in this diagram you can see the male gametes get traveled within the pollen tube. They are actually non-motile. They are get traveled by the content. They don't have any motile organ. So such the movement is said to be siphonogamy, and here you can see the porogamous entry, that is the porogamy, and they will reach here up to the synergids. Okay. So in double fertilization, after entry of the male gametes, one of the male gamete unites with egg which is present here, this one, and another male gamete will unite or fuse with PEN, that is primary endosperm nucleus, and hence it is called as double fertilization. Here you can see the label secondary nucleus plus male gamete, means nothing but the fusion of secondary nucleus plus male gamete, this one, and here syngamy or first fertilization is nothing but male gamete is fused with egg. So let us see process in details. In double fertilization, there are two types of fertilization. Already I have shown in diagram. One is first fertilization, which is also called as syngamy. Another one is second fertilization, which is called as triple fusion. So one of the male gamete, as I have shown earlier, unites with or fuses with haploid female gamete, that is nothing but egg, and it will produce a diploid zygote. And this zygote further develops into embryo. Such type of syngamy is nothing but a generative fertilization. It is nothing but generative fertilization because it will develop into embryo. And second fertilization, that is nothing but one of the male gamete, one get fused with egg. Now there is another one that will fuses with primary endosperm nucleus, where there are two nuclei. So both are again haploid. That two nuclei plus one male gamete, which is again also haploid, will form triploid endosperm. It will form triploid endosperm. And this fusion is nothing but vegetative fertilization, because further it will form the endosperm. So let us see significance of double fertilization. It is unique feature of angiosperm, already I have told you. It ensures the parent plant invests a seed with a food store. The food is required within seed for their nourishment. That's why due to double fertilization, the food is stored. Now the diploid zygote, which is developed from syngamy, develops into new plant by formation of embryo or through formation of embryo. The next, there is the triploid primary endosperm nucleus, develops into nutritive endosperm tissue. This is the next significance. Now, due to diploid condition, it maintains the diploid condition throughout or through next generation or carried to next generation, and hence it also avoids polyembryony. So let us see how the endosperm is developed or development of endosperm. Triploid primary endosperm nucleus repeatedly divides by mitotic division to form a nutritive tissue. That tissue is called as endosperm. This is the four fertilization changes which are taking place within the ovule. So these endosperm as well as embryo get developed simultaneously. There are three types of endosperm. One is nuclear endosperm, nuclear type of endosperm. Another is cellular, and third one is halobial. So first one, that is the nuclear type. It is the most common type of endosperm found in almost 161 angiosperm families. So primary endosperm nucleus, that is PN, repeatedly divides by mitotic division, and it will form large number of free nuclei like this. In diagram you can see large number of free nuclei are formed. Here also you can form large number of free nuclei are formed due to the repeated mitotic divisions. And at the center there is formation of vacuole, which results into the pushing of these nuclei towards periphery, and later on it will form a cell wall surrounding the nuclei, but this formation of cell wall is incomplete. Okay, it is incomplete. Here you can see the wall formation remains incomplete. So such endosperm is found in wheat, sunflower, coconut. In coconut, you are well known that there is a coconut milk. There is a liquid, ज्याला आपण नारळाचं पाणी म्हणतो. That coconut milk is nothing but endosperm. Which type of endosperm? Nuclear type of endosperm. Next type is cellular type. So here what happens? Again the same thing, same procedure. PN, that is the primary endosperm nucleus, that is again having the triploid condition. It will divide and redivide first by nuclear, but immediately it is followed by wall formation. So wall formation starts from the beginning, okay, so which results into such type of endosperm, which is called a cellular type of endosperm, and it is observed in almost 72 families of dicots. For example, in balsam, petunia, adamsa, etc. Third type of endosperm is nothing but halobial type of endosperm, which is only found in halobials order of monocotyledon. Here what happens? First division of PN will take place, which is again followed by formation of transverse wall, this wall, which divides into two cells. One is larger, another one is smaller. Smaller is present towards chalaza, larger is present towards micropylori end. Now nuclei in each cell divides, and same thing is here in this larger cell. You can see the nuclear division. Here also the nuclear division in smaller cell. So such type of endosperm is called as halobial type of endosperm, which is forming as spudulus. Another type of endosperm is also found, that is in males, mosaic endosperm. So here what happens? Endosperm contains two different types of tissue. Here you can see two different types of tissue in endosperm. This is one type, this is another type. So which show the patches of two different colors, which give the appearance like mosaic, and that's why it is called as mosaic endosperm. Now let us see development of embryo, how the embryo get developed. The process of development of zygote into an embryo is called embryogenesis. This embryo is developed at the micropylar end of embryo sac. So what happens? Zygote divides into two-celled proembryo. Larger cell which is present towards micropyle is said to be basal or suspensor cell, suspensor initial cell. Smaller cell which is present towards chalazal end is said to be terminal or embryonal initial cell. Let us see in diagram. So here you can see the larger one, which is called as suspensor cell. This one is called as embryonal initial. Okay, this one suspensor. Now what happens? Suspensor cell further divides by transverse divisions and form 6 to 10 cells like this. 6 to 10 cells are formed here. Okay, here also you can see the same thing. Okay. The larger one, which is present towards micropylori end, this one is called as posterior, and lowermost, this one is called as hypophysis. This suspension pushes the embryo into the endosperm. So this one is the micropylorate, this one is the chalazoline. So it pushes like this from this direction. Okay. Now the embryonal initial, which is present here, this one, embryonal initials, further divides into 3 successive mitotic divisions and results into formation of 8 cells, and the structure is called as octant, as there are 8 cells like this. Here you can see 4, again it will undergo into mitodivision to form octant. So thus it forms 2 tiers. Here you can see 2 tiers, outer one, inner, or you can say lower and upper. So lower tier of 4 cells of octant give rise to hypopotyle and radicle. So it will lead into the formation of hypopotyle and radicle. Upper tier, which is present outside, upper tier will lead into the formation of plumule and cotyledons. Here you can see cotyledons. Okay, this is the plumule, this is the cotyledon, which is formed by the upper tier, and here lower tier you can see hypopotyle radicle, which is formed by the lower tier. Okay, so these are the parts of, you can say, embryo. Embryo development is similar in both dipods and monopods up to the octant stage. Same procedure, same steps. In monopod embryo, single cotyledon occupies terminal position and plumule is lateral. Single shield-shaped cotyledon is called as a scutellum. So it will work as a shield shape. And protective sheath of plumule, here it is called as polyptile, and that of the radicle is also covered by sheath called as coleorhiza. Ovule is transformed into seed and ovary into fruit. You are known about this. Ovule will further form the seed and ovary will form the fruit. So, thank you.